Synthetic biology treats biology as an engineering discipline. It focuses on the rational, systematic design of biologically based systems that display functions not found in nature. This chart tracks global newspaper articles about synthetic biology since 2004. Note the steep, cumulative upward curve. Early breakthroughs triggered this sudden explosion of public interest. As media coverage increases, the discussion often prioritizes science fiction scenarios over technical realities. Public perception begins to drift away from the actual work happening in the lab. Physical research is actually conducted in highly controlled, sterile Level 1 laboratory environments. These spaces are defined by strict containment protocols and methodical safety practices. Scientists in these environments work with well-understood, apathogenic organisms like baker's yeast. They are not creating uncontrollable life forms. They are modifying standard biological hosts. This gap between the public's image of designed life and the methodical reality of the lab creates a specific tension for anyone attempting to write policy. This is the Collingridge dilemma, the fundamental challenge for governing emerging technology. It maps the relationship between our power to change a technology and our understanding of its effects. During the early stages, regulators have the most power to shape how a technology develops. However, they lack the concrete physical data required to write evidence-based rules. By the time researchers have enough empirical data to understand the risks, the technology is already integrated into the economy. The infrastructure is set and difficult to restrict. Biotechnology development moves at a pace that intensifies this problem. Biological systems scale and replicate through growth, which differs from the deployment of digital software. To solve this dilemma, regulators must separate short-term biological risks from long-term sociological expectations. Effective governance requires focusing on what is physically happening in the lab right now. Success depends on mastering the timeline of empirical data. It is a matter of waiting for the science to provide evidence before drafting permanent restrictions. Evaluating short-term risk requires a map of how these systems will be built over the next decade. Researchers generally follow two distinct paths. The top-down method involves taking an existing organism and removing every known essential gene. This reduces the genome to the bare minimum required for survival. This process creates a minimal genome organism. It functions as a blank slate chassis, a highly efficient biological tool optimized for a specific task, such as producing a medicine. The bottom-up method works in the opposite direction. Researchers use standardized fragments of DNA to build novel metabolic pathways in a precise, linear sequence. This assembly requires the designer to know the exact function of every genetic part. These components cannot organize themselves. They must be put together according to a specific plan. Both methods rely on a predetermined engineering design. This removes the random mutations and unpredictable changes associated with natural evolution. Because these systems are rationally designed from known parts, they lack the unknown elements found in wild organisms. This makes their behavior more predictable and their risks easier to calculate. While it is often assumed that new technologies need new laws, the existing frameworks for genetic modification are already suited to synthetic biology. The physical risks remain the same. The primary safeguard is biological containment. Synthetic organisms are engineered to be dependent on specific nutrients that do not exist in the natural environment. Minimal genome organisms are physically fragile. They are designed to thrive only in highly specific laboratory media, making them unable to survive or replicate if they were to leave the lab. Researchers also use host organisms like E. coli K12, which have been studied for decades. These strains are non-pathogenic and do not pose an infection threat to humans.
There is a cost to overregulation. Assigning every synthetic biology project to the highest containment level creates financial barriers that can stop innovation before it produces any benefits. Short-term synthetic biology is physically contained and biologically weak. Under current protocols, the escape of an artificial pathogen is prevented by the basic biology of the organisms themselves. If the lab-based risks are manageable, we have to ask why there is such strong public demand for new, restrictive regulations. Science relies on funding, and funding often follows expectations. Scientists describe the potential future benefits of their work to secure resources and create a space where long-term research can happen. This is Gartner's technology hype cycle. It shows how the visibility of an innovation changes over time, regardless of its actual maturity. During the peak of inflated expectations, promises of future breakthroughs lead to intense media sensationalism. This is where the gap between lab reality and public perception is widest. At this stage, public discussion focuses on ethical fears, such as whether life can be reduced to a set of DNA instructions. These philosophical questions overshadow the evaluation of physical risk. Wynne Semius's policy cycle illustrates how governments respond to this visibility. It tracks political importance against the amount of disagreement in society. Note that the policy formulation phase begins right when public disagreement is at its highest. Policy is often drafted based on the peak of the hype, long before the technology is actually ready for the market. Globalized research makes this even more complex. National policy is limited by international trade agreements, meaning one country cannot easily regulate a technology that is being developed worldwide. This is the Collingridge dilemma in action. Governments feel forced to regulate the hype surrounding a technology before they have the physical data to regulate the technology itself. Regulators must also prepare for more complex, long-term projects. In the future, researchers may engineer systems with hundreds of genes, where every interaction cannot be predicted in advance. When facing these unknowns, governance relies on the precautionary principle. This is the technical fail-safe for biotechnology. The rule is simple. If the risks of a synthetic organism cannot be adequately assessed, the project is automatically restricted to the highest level of physical containment. This is a staircase progression. An organism only moves toward a lower containment level once researchers verify a rigorous set of safety data at each previous stage. Scale and environmental exposure only increase after empirical evidence from the previous, highly contained step is confirmed. This mechanism protects the public while giving regulators the time needed for the science to mature. It ensures that safety is based on evidence, not on the initial hype of the technology. Modern governance follows two tracks. It enforces strict physical containment in the lab while monitoring the sociological hype in the public sphere. During the peak of inflated expectations, the government's role is to facilitate ethical debate and map societal boundaries. It is not the time to draft restrictive laws based on theoretical fears. Eventually, the technology will move past the trough of disillusionment. It will reach a plateau where synthetic biology produces stable, economically viable applications. The development of complex biological machines is a slow, incremental process. Each engineering step provides the time needed for risk frameworks to adjust to the new data. Synthetic biology is an evolution of genetic engineering. It is a continuation of our ability to work with biological systems, not a sudden departure from them. By understanding the physical limits of biology and the sociological mechanics of the hype cycle, we can guide these technologies out of the realm of panic and into a period of